Should we finish 13 and kind of start 14?
Yeah, we can. And I was looking over. So you guys are the big winners today, by the way.
With the quiz?
Yeah. I gotta, like I said, I got all my stupid EMT quizzes done tonight. So I've been studying this stuff and going over my stuff. I have 2 exams tomorrow and 2 other classes. So I'm just trying to, as much as I'd love to go to the movie tonight, I'm not going to be doing that. So that's all right.
You can reward yourself tomorrow.
What was that?
You can reward yourself tomorrow.
Oh, it won't be until Saturday. It won't be until Saturday or Sunday or Friday or something. So I'll be tired tomorrow. There's no way I'm going to the movie tomorrow. And I have to go back to work tomorrow night. I mean, I start work at midnight tomorrow, so it'll be okay. It's fine. I'm not worried about it. Okay. So
do you know?
I'm not really sure.
I think we went over heat and steam.
How did you guys get done with that so fast today?
Oh my gosh, I don't know. It was so easy.
Well, no, it wasn't hard. I was just like, oh my gosh, like how are they getting through this so doggone fast?
Well,
it's okay. Hey, what's up?
We were
what are you guys talking about?
Oh,
well, I was just saying how you guys got to do the lab so fast today.
Oh, because Laura knew what she was doing and helped me. Basically.
Okay. Well, no, that's fine. I just was like, man, you guys got done so doggone fast. Okay, so here I'm on.
I just did before this. I did the next discussion that's due on the 25th class.
Mm-hmm.
And
it's is it up? Is it online?
Yeah, it's just a documentary. It's like 55 minutes, and it's super easy. You just answer some questions.
Okay, hold on. Let me see if I can bring it up because
those aren't for credit, right? Those are just for the tests.
Yeah.
Okay.
Yeah, yeah. Hold on. Yeah, I'm just saying that I know I got caught a little bit with my pants down on the last test because she asked quite a few questions from those that I was like, you know what I mean?
Yeah.
I was like, I refuse to do that again. I'm not going to ever do that again.
But
so is that under?
We don't have any of those for this next test.
Yeah, no, but is it antibiotic resistance, Addison? Is that the one?
Yes.
Yeah.
Okay, yeah. There's a worksheet that goes with it, right?
Yeah.
Okay.
Super easy.
So you watch the video and then you do the worksheet. Okay. Got it. Okay, I'm gonna print this out right now. I'm going to save it in my files and I am going to print this out. I am not going to get caught with not doing these again before she talks about them ever again. So this is going to be in exam 3, right, Addison?
I believe so.
Okay. I'm just sorry, ladies. I want to get calibrated because it just feels like there's a lot of stuff going on right now with this class. And I just don't want to get behind on any of this crap. Okay. So where were we at on this? So, okay. So like I said, we got 3 things coming up, right? We got our midterm in the lab next Wednesday, and we have the exam and the class on Wednesday, right? They're both the same day, which really isn't that bad because a lot of it's going to be reviewing for the other one. You know what I mean?
Mm-hmm.
I mean, yeah, you can get all bent out of shape about it, but at the end of the day, it's all going to be the same stuff. You know what I mean? So my question is so we've got basically today, tomorrow, Friday, Saturday, and I'm just recommending that we just don't even study on Sundays, you guys. It just seems like a real pain in the butt to do that. And I like to have a day off to do. What was that?
It's nice to have a day off.
Yeah. Well, and I'm not. Again, it is what it is. It just seems like the weekends is really truncated. It's hard to work that time together. So I'm just recommending that we go Friday at 3. You know, whoever can do it. We go Saturday at 1 with whoever can do it. That lets all of us, because like I have a Zumba class that I want to go to on Saturday mornings and stuff. So like I'm like everybody else. I got other things I want to do as well during, you know, and so but 1 o'clock just seems to be a time that's open on Saturdays.
Yeah.
Unless I'm confused. So
no, my work schedule is just different every week.
Yeah, basically. No, and like so for you, it just okay. So are we all in agreement that like Monday through Thursday, 6 o'clock kind of works for everybody, and then on Friday at 3, and then Saturday at 1. I just want to kind of get, because I'm going to put in my schedule and start making like actual announcements, you know what I mean, and actually really do it.
Mm-hmm.
Thing is,
So
there's going to be 2 or 3 days a week that I won't be
yeah,
able to do that.
no, that's fine. And I know, Addison, with your work schedule, it's going to jump around a little bit, but I mean, are you comfortable with that? And then if we need to, you know, like I said, you got me and Laura. If we need to navigate it, whether it's study in the morning or study in the evening, I think between Laura and I, you can find somebody else to study with if you need to. That makes sense.
Yeah.
Okay. Other questions. So this is more for Addison than for you, Laura. So we go and I'm talking about meeting with the teacher every Tuesday and Thursday. And right now, I just don't think it's very effective. And so you went the one time that I went and met with her, you were there. So is there a way that we can prep that and get ready and be more effective with her? Does that make sense what I'm asking?
I feel like she just doesn't really, I don't know. It was just weird, I felt like.
Well, yeah, and I'm gonna send her an email and just say, Liz, I'm gonna talk to her about it. But I just wanted to feel you out because you were actually there. And so I guess the better question, maybe Addison, for you is if we were gonna set up office hours with her, like we're kind of trying to do, how would you like to see that going, and what would be the most effective for you? And what do you feel her role would be in that? Does that make sense what I'm asking? Is that a little bit better way to ask that?
Yeah. I don't really know, honestly.
You know, I'm literally considering asking her, like, if I bring my computer and set it up, actually having her do like mini lectures with us, if that makes sense.
Yeah,
like
I feel
actually
like
have
that
her
would
introduce
be
the
better.
material to us quicker. What was that?
I feel like that would be better too.
Okay. Oh, I'm going to
Really
talk to her,
say
and I'm thinking if we did maybe 15, 20, 25, 30 minutes with her, whatever she's willing to do, but just literally start going over the materials sooner. Let's start working through it and having her answer our questions and whatnot. And that's pretty much it. I just wanted to feel you guys out and stuff, because right now, again, I just don't feel like I'm be. And again, I've had some things drawing my time with the EMT class and stuff, which is kind of gone now. I mean, I still have to test for it, but now I'm just done studying for it. I'm ready to just schedule my damn exam and be done. But yeah, so for you guys, I just want to make sure. I think you guys are definitely getting the response. I mean, you guys definitely both scored well on the quiz today. I take it.
Yeah, I got 10 out of 12.
I boloed the shit out of it. I'm not sure why I did that. I second guessed 3 of the answers that I did and whatnot. And so I guess this is what I'm going to ask of you guys that would help me. I'm happy sponsoring this. I'm happy running it. But again, I'm using this time to recalibrate, to get a focus. And I'm asking you guys that what I want to start doing with our study group is I have no problem just going through slides, but I would like to start getting questions. So like, I don't know, like splitting the slides up, and, you know, on each slide, there's at least one question, whether it's generated from her or that each of us could generate. I know that takes a little bit of time to do that, but I would benefit by getting together on this. And so Laura has 10 or 15, 20 questions that she's created from chapter 7. You're going to learn it better, Laura, and you're going to run us through your questions. And I think that's a more productive way. And then, like, for me, it forces me to go to chapter 9, if that's what I'm asked to do for the group, and generate 10 or 15, 20 questions. So we start getting questions. We start seeing things as questions as opposed to red words on a page and a slide. Does that make sense? I really need to start saying things in question format. It's going to force me to think, and then you guys can talk me through your questions as you ask them. If I answer them wrong, then I have my peers to kind of walk me through the mindset, and I can start to see question format of this stuff, and it's going to make me think a little bit differently. Is that okay? I thought this out today as I left from our lab, and I think that's the next level of where we need to go, is that we need to do what we're doing. I think this is effective. Reviewing slides together and going through stuff is really, really effective, and I think it's working. I just need to calm down and let things work for me the same way that's working for you guys. And by your own admission, Laura, you had some extra help, right? You and your husband are studying together.
Yeah.
So how many hours a week extra do you think you're getting? I'm just kind of curious. I wanted to hear from you. In addition to what we're doing, how many extra hours are you getting from him?
Last night was the only time we did it, and it was like 2 hours.
Okay. So for you, that clearly that extra 2 hours focus study with your husband really helped. I mean, that's a good way to put it.
me explaining it to him. So I had him open up to the beginning of chapter 8 and then tell me like the first like topic on the slide, and then I would teach him
everything from the slide.
Yeah.
Okay. Well, I appreciate your insights, because I want to, yeah.
I think maybe it would be less stressful for you if maybe we divided up the slides and each of us, like we would rotate, like maybe the first 9 slides you would teach us, and then the next 9 I would do, and then the next 9 Addison would do. That way we're like diving into it a little bit more, but it's not all on you to go through it.
Well, yeah, no. And I think the other day we did that. I think the other day when you guys took like 7 and 8 or whatever we did, that was really what happened. And like I said, the next thing for me, as I was thinking about today when I left, for me, the next level is seeing questions, not just pounding out the slides that she's already showed, but like, okay, so I'm looking at steam sterilization, right? There are at least 3 or 4 questions on this that we could like, so, okay, so steam sterilization. So, okay, what is the, like a question that I would ask you guys is, what is the temperature, the pressure, and time that we have to subject things to in order for steam sterilization to be appropriate, right? And then you have, and then I could do it multiple choice, or I could make it fill in the blank. That makes sense. But it forces you guys to start seeing that there's more than just, oh, autoclave and steam under pressure, and we kind of sort of look at it and we move on, right? And whatnot. So that I think that's where I'm going with it. For me, I need to start doing that. And like today, it was like the very last thing on this slide, endospores was the answer to that one question.
Well,
and I just don't. What was that?
It's the middle one, where sterilization because bacterial endospores killed.
Well, yeah, and then but I'm saying here at the bottom too, it says endospores down here, next to stereo is endospores.
Blank is what the answer was.
Yeah. Okay. Yeah. No. And like I said, but there's at least 3 or 4 questions on each one of these slides. Like if I click it over, and then like, okay, having a picture with all crap pulled off of it, you know what I'm saying? And talk about some of these different, not that. And then like here, like pasteurization, like, okay, showing this picture with all this gone, and then having, one of us kind of talk through these bullet points. Okay, it's heating, below boiling point. I think you got a really good job this other day. It does not sterilize, but does kill pathogen, because it's this up and down, slow, whatever. I'm just throwing that out there, ladies. I was thinking about ways that we could improve this. I didn't want to take the whole time talking about it, but again, all I'm asking you guys to do in the next day is to kind of get the juices flowing. Think about what you think is working well, and think about what we could do to improve this, and then be willing to come to the table and offer that. And I think, yeah, whether it's take 9 slides to teach them or whatnot, because let me ask you guys a question, because I was keeping track today. How many total slides did she cover today in class? So she started at slide number, I was kind of keeping track. So she started on slide number
8.
She covered how many total slides?
She started at 8 and left off at 56.
Okay, so that answers that question. So she's covering,
about
give or take,
slides.
About 50 slides a day. So if there's 3 of us, divide 50 by 3. So that's the level. That's the speed at which we have to go. We got to be covering 50, 60 slides in our own study group on our own. So like where I'm at with it is, okay, so we have class 2 days a week. There's 7 days of class. I mean, there's 7 days in the week, right? Excluding Sunday. So that means for every day in class, we have 1.3 days out of class that we're on our own, right? Well, let me rephrase that. Including the days. So basically we have 2 days in class, and we meet 6 days a week to practice. So that gives us basically 3 days to work on our own. And so if you look at that, like, so what you're saying, Laura, so if she teaches 50 slides a day in class, and each of us takes like 18 slides or whatever, a third of that is. And so the first day I take the first 9 that she covered, or the first, 18, you take the second 18, Addison takes the third 18. And then the next day I take your slides, you take Addison's, and she takes mine. And you cross your bridge however you want. But by the time we hit the third day, each one of us had to teach everybody else's stuff and we're seeing it all again. And like for me, I know for me, I'm going to start generating questions from the slides where maybe Addison just reviews the slides like what we already have, where Laura, you do something else. If that makes sense. But that gives each person a chance to be a little bit more innovative and to cross it a different way. Again, I'm just thinking about that. I'm doing the math of if she covers 50 slides a day and we have 3 slides, we have 3 days between each lectures, give or take, to cover that material. Then again, that means we could divide it. And then each day we could each cover the other person's section to the next lecture and then move on. And then that way we've taught it all, we've seen it all, and we've seen it 3 more times in our study groups. And we've all had a chance to teach it to each other. And it gives you the innovative process to actually break your slides up and look at them and maybe make flashcards on Quizlet or, you know what I mean? Is what I'm saying? Whatever you want to do. That's it. I just wanted to show you guys some ideas that I was having, ways to be more innovative and to make this a little bit more effective for all of us. Okay, I'm done. Is that cool? Sorry, I didn't mean to take the first 15 minutes to have that discussion. I just wanted to touch base with you guys and kind of let you know what my thoughts were. That I want to get this to where we're all getting A's all the time and we're really confident with this all the time. It's not we're ever shocked and we're having to study 3 hours the night before an exam with our husband because we feel like we need to do that. It's like, hey, the pacing of what we're doing is allowing us to get through this and we're confident all the time, no matter what people drop in our lap, we're ready to just go test. Okay, that's it. Thanks, ladies. I didn't mean to talk about that long. Okay, where are we at? What do we do? What are we doing tonight? What are we doing tonight?
Finishing 13.
So where are we at on 13? Do you remember? Damn it.
I think
I got to turn this stupid group off right now.
I think we would just finish dry heat. Maybe.
Okay, hold on. I got to shut these stupid. Okay. For the next 24 hours, I won't be receiving any. Okay, I just shut that stupid. I keep getting hit with stupid message on my Facebook. Okay, so where are we at? Slide number 13. What number is it, Laura? Do you know?
20.
Oh, dry heat. It's 21. I'm right here. 21, dry heat.
I think, didn't we do that or not?
Oh, let's just start here.
Okay.
I can't remember. Let's just start.
I think we did refrigeration, but
we did.
Let's just start here.
I don't think it hurts us to start here, you guys. It's okay. I think we're going to be okay doing this. Okay, so dry heat. Hey, ladies, is it okay if we step back to the front really quick and go through the definitions really fast on slides number 3 and 4? Can we do that really fast?
Sure.
I just want to hit those really quick again because there was quite a bit today on that stuff. It just seemed like there was a lot there.
Well, I'm mad because I didn't think she was going to do anything from 13. And the 2 I missed were from 13.
Okay. Like I said, let's just hit these definitions really quick. So remember, there were 2 parts to this, Laura. You and I went through this last night. I don't know if Addison was here. So there's basically 6 definitions, but the definitions are broken up into 2 classes. And the classes are what? What are the 2 classes of the definitions? It has to do with this word right here. Inanimate and organics, right? So the first 3 words, sterilization, disinfection, and sanitation, all have to do with what stuff? What do we do that to?
Bacteria?
Well, no. So sterilization, look at the red words and sterilization, disinfectant, and sanitation. Look for the common word. Object. Yeah, those are all inanimate object stuff. So like for me, I'm going to make a note card now with these 3 terms and just the inanimate object piece on it because I know what they mean. I just get confused about where they fall. So what's the difference between sterilization and sanitation?
It just decreases sanitation growth and sterilization of all.
Yeah. So like sterilization really is like the granddaddy of them all, right? It kills everything. It doesn't just kill the bacterial cells. Like sterilization would kill good living human cells too, right? That's the kind of stuff that when you put it on your skin, it burns your skin because it's actually killing your cells and the bad stuff, right? And that's why we don't use it on organic material because it just kills everything.
Peroxide?
Yeah, like that's a great example. Hydrogen peroxide kills living cells and pathogens, and it destroys everything. And that's why we really resistant to use it on living objects, right? Okay, so and then disinfection. So basically, on a scale, the most devastating that kills the most stuff to the least devastating that doesn't kill, that kills the least amount of stuff. How would you rate these three words from highest to lowest? Sorry, Addison, I didn't hear you. Sanitation, what? Sterilization, what?
Is the worst. It like kills all of them.
Okay, and then so what would be the next one?
Disinfecting.
Yeah.
Yeah, so disinfecting would be the next highest because there's still some killing going on, right? There's still some destruction here. Where does sanitation require that we kill anything?
No.
No, it either decreases its, like this. Remember, it's the side versus the what was the other one? Remember, their side versus the one that just inhibits growth. It doesn't actually kill them. And so that's what's going on here. This one here, sterilization is killing everything. Disinfecting is killing targeted pathogens on inanimate objects, right? And then sanitation, all we're really doing is just making it safe. We're not actually killing anything. Okay, and then the next one is, again, we had this one here, and this one caught me off guard too. I was able to answer it kind of, but I wasn't able to give her the answer I think she was really looking for, which is it kills or inhibits pathogenics in living tissue. Okay, and so antiseptic, degerming, and chemotherapy all have to do with living tissue, right? It all has to do with like us and, you know, animals and plants and stuff. So antiseptic would be the highest on this, right? Because there's a killing agent going on, right? And it's killing pathogens on living tissues. The next one is degerming, right? So, or actually, excuse me, on the level of attack, antiseptic is the highest, right? Then it goes chemotherapy because, see, it's using chemicals to internally kill stuff or inhibit growth within the host tissue. And the last one is degerming. And I was teasing Laura the other day that degerming is like making her husband wash the dishes. He's actually not killing stuff. He's just removing food and refuse particles from the dishes, and it's a mechanical wash. It's like washing our hands with soap. We're not actually killing anything. We're removing the stuff through mechanical means. And so that would be the least inhibited because it doesn't kill anything. It just removes it from our skin, and then doesn't hurt us that way. Okay, is that helpful to do that?
Yeah.
Okay, I just wanted to hit those because she did hit those, and my gut tells me we're going to see those again. Okay, going back to heat, we're going to go back to the dry heat. Okay, here we go, dry heat. So what does it say? So dry heat is less effective, right? But it requires higher temperatures and a longer exposure time. So let's do a quick review. So on wet heat, on our wet heat that we just talked about, so there were 3 conditions that had to be met for wet heat to kill and to sanitize, like medical instruments, for example. What were they? They were temperature. So what temperature did it have to be at?
Like 121 or something.
It was, yeah, it was like 100. It was like 100. I think it was 150 or 120, something like that. Let's look at it. I'm gonna look it up to make sure. But see how easy it is to forget that. That's why I'm kind of like, I was looking at this earlier and I was like, man, there's just some pieces here that she kind of caught us with her pants down today a little bit with some of this. And so yeah, so it was so for wet heat, the 3 conditions that have to be met in order for wet heat to work. You are correct, Laura. It's 121 at 15 pounds of pressure and for 15 minutes. So 121 at 15 at 15. Okay. Good. So dry heat, what are the 2 conditions that must be met for dry heat then? The pressure is gone now. The pressure part of this is gone. So it's 170 degrees Celsius for 2 hours. And there is no pressure component for dry heat. And then the last component for wet and dry heat is what they'll actually work on. So what kind of things do we put inside of a steam sterilization or an autoclave? What do we put in there to sterilize?
Plastic.
It doesn't actually say. So in the dry heat, we put things like glassware, powders, and oils in there.
The one
and then
where it goes through a box.
What was that?
Weren't you and I talking about something that goes through a box? Or was
so
that
that's radiation. That's a particle accelerator that does that.
Okay.
So sterile, so autoclaves or steam heating, that's for like medical instruments, like things that are metal, right? So we can put like medical instruments, like scalpels, that kind of stuff through there. And so that kind of stuff's what we would put through an autoclave. But so for example, would we be able to put like linens from beds through an autoclave?
No.
Or do we put those through dry heat?
Dry heat.
Dry heat.
Yeah, well, and technically we don't put bed linens through dry heat either. We actually wash them, and so instead of using dry and sterile heat and wet heat to clean those, we use a chemical process, right? By adding detergents and chemicals to bed linens at a hospital. So we're going to use that degerming first by washing them, and then we're going to expose them to chemicals. So we get all the big particles of blood and human tissue and stuff off of them through washing them or degerming them. And then once that's done, then we put them through a sterilization process that doesn't require any of this stuff. We put them through like chemicals, right? So we may put them through some kind of acid, and then we clean that acid off, and then we dry them again, and then they're at that point they're ready to be used for humans, in a bed again at the hospital. Okay, but just so what are the key things for dry heat? Who wants to go through them real quick?
It's 170 degrees Celsius.
Yep.
It requires higher temperatures, like the 170, opposed
Yep.
to like the 121. It's used for things damaged by moist heat, so we would use glassware, powders, and oil.
Okay, and is it more or less effective than autoclave or wet heat?
Less of.
Yeah, it's not as effective. It just doesn't work as well. Okay, any questions, guys? Ready to move on?
Yep.
There's two kinds of dry heat. What are they?
Flaming or incineration.
Yeah. So flaming, what would be a good example of flaming that we use in the lab every week?
The Bunsen burner.
Yep, we use the old Bunsen burner. And then incineration. What's incineration? Does anybody know?
Like they put it in an incinerator?
Well, yeah, you can't don't try to use the same word to explain it. So an incinerator is just like basically we put stuff through a room that's really, really hot, but it doesn't always have to have flames. Does that make sense? So flames.
Yeah.
So like here in the picture right here, they showed this guy putting his inoculation loop through this wire grate, and what that's done is that stuff gets really, really hot, and so the ambient temperature inside the tube inside here gets very, very, very hot, probably up to a couple hundred degrees, but there's no flame present. Does that make sense? There's not actually a flame in there that's present. It's just it's like putting it inside of an oven. It's basically a little dry heat oven for an inoculating loop, but there's no flame present. Where with a Bunsen burner, what are we using to do that?
A flame.
Yeah, an actual fire, right? There's an actual
flame.
flame involved, and that's the difference between flame and incineration. Incineration does not require a flame. It just requires a really, really hot space to do it. Okay, refrigeration. Who's doing this one? Who wants to take this one?
Me.
Take this one, Laura.
So temperature 0 to 7 degrees Celsius, the microorganisms can't reproduce. And the exception is listeria. And then
So can you explain that? What's going on with listeria and why it's an exception to this?
I have no idea.
Can it actually reproduce in these cold temperatures? That's the question that I'm asking.
I guess so.
I think it can.
Is that what she said? Okay, no, that's why I'm asking. I know that we're saying, hey, listeria is the exception, but I'm not entirely sure why it's the exception. And I just thought I'd ask. So maybe I'm going to make a note that when we see when I see our teacher again, I'm going to ask her on that one. Say, why is listeria the exception? And then if she can tell us or whatever. Okay, keep going, Laura. Sorry. Thank you.
No, you're good. And then most things can survive in temperatures less than 0 degrees Celsius, but they don't grow.
Yeah, they just go dormant. Okay. So Addison, do you want to take desiccation? What's going on with desiccation?
It's when they like remove the water, right? And they like dry it
remove
out.
water.
So simple desiccation is like dried fruit or beef jerky. The life of the freeze drying, it could be like freezing your food or vaccine, and then you suck out the water.
Okay, so you freeze it and then suck the water out. Is that what you're saying for dry freezing? So you
I
freeze it,
believe
then you
so.
dry it. Okay, thank you. And you're saying that we do that for vaccines and certain kinds of foods?
And yeah.
Okay. So like space shuttle food.
Mm-hmm.
Okay. Okay.
And then simple would be like dried
Yeah,
fruit.
like beef jerky and stuff.
Mm-hmm.
Okay, here we go. Desiccating. Sorry, guys. Sorry, ladies. We're desecrating by reducing water activity. So situations that we do this, so we have high and high salt and sugar concentrations to create a hypertonic environment, right? Because we're trying to pull water out of it, or we're trying to push water in, or is it hypotonic? We're trying to pull water out of the stuff, so it would be hypotonic environment that we want to pull stuff out.
No.
Is it hypertonic?
It's hyper.
It's
Okay. So we place our food inside of a hypertonic environment, and because the concentration of the salt or sugar is higher on the outside, it pulls all that water from our cells, our whatever we're trying to preserve, and that's what's going on. So what happens to the?
It would shrivel and do plasma myelolysis.
Yeah, that can happen, right? Do we want to do that to our food? Do we want to destroy the plasma? What are we trying to do that to? We're trying to do it just to the microbial cells, right? We want to keep the food good. We just want to destroy the microbes, not the food. So what happens is you're right, Addison. It makes for plasma. What was it? Plasmolysis. Plasmolysis. And Laura, what's plasmolysis?
Plasmolysis is when you the water gets sucked out of the cell, and the edges of the membrane detach from the cell wall, or some of them do, not all of them.
Yeah, does the whole thing get detached, or just there are parts that stay connected, right?
Yeah.
And when those connected parts get under too much stress, what do?
They tear off.
Yeah, they tear. That's exactly right. So we have just regular lysis is what? What's regular lysis? So osmotic lysis or cell lysis. When we start pumping the cells too full of water, they rupture because they
explode.
Yeah, they get too big and they explode, right? And then plasmolysis is where we suck the water out, and the plasma membrane that's still attached to the cell wall, it stays attached, but as it gets shrinks, the part that's not attracted or not attached to the cell wall, it starts to tear because there's parts of it that stay attached no matter what, because it wants to be attached to that cell wall. So that plasma membrane gets shrunk down more and more and more. Eventually it just can't handle it and it ruptures and tears open, which is obviously not good for these guys. Okay, radiation's like my absolute favorite. I love radiation. This is so freaking cool. So for those of you who are going to be taking bioorganic chemistry in the future, guess what? You're going to be talking about radiation in that class. So have you two taken bioorganic chemistry yet?
No, but we're probably going to take it together.
Yeah, just know there's a whole unit about radiation and DNA and about this kind of stuff. So the better you learn this now.
Cancer center.
Sorry, go ahead, Laura.
I work at a cancer center, and so it's fascinating to me too.
There you go. Okay. So we kill microbes by damaging the DNA. And really what's going on is it's basically like shooting bullets at it. So you shoot these gamma X-rays and high energy photons and electrons into it, and UV light into the DNA structure, into the cell structure. And what it does is it just cuts it up. You know, those high energy, and that's one of the problems with like nuclear bombs and stuff. It's not the bomb blast that gets you, it's this radiation that kills a lot more people, because it cuts their DNA and causes cancer and birth defects and stuff. But this radiation is extremely effective at killing bacteria as well.
What was that on the quiz today? Oh,
it was non-ionizing radiation or UV light.
Timing dimers, that's what it was.
Yeah, and it's on here somewhere.
Yeah, it's like 3 slides. Yeah, you were just go back.
Yeah, it's coming up. Okay, so that's it. So what's the difference between non-ionizing radiation and non-ionizing radiation, ladies? Do you guys know the difference?
The non is just the
ionizing
UV light.
Sorry.
I just said non-ionization. It uses UV light.
Yeah, what it really comes down to is the speed at which the particles move and what they can penetrate through. Does that make sense? So gamma rays, if you look at the scale here, right up here at the top, gamma rays are moving at very, very, very high energy. And so they have the ability to penetrate deeply into a lot of things. And then, so like, for example, in order to shield for gamma rays, like, for example, so have you guys ever been in for x-rays for your teeth or for bones? What do they put over you to protect you?
Oh, what's it called?
They
Lead.
put a big heavy lead vest over you, right? And that's to protect you from the x-rays. And so like gamma rays, it takes a couple of feet of cement and lead to keep from getting the gamma rays through. That's why we shelter against like a nuclear attack or radiation attack by going underground, because the ground gives us a lot of protection. Okay. So gamma radiation is the highest energy. Then there's X-rays, and then there's high energy electrons, beams, and then there's UV light. Now, let me ask you guys a question. What is something that happens to us outside in the summertime when we stand outside? We're getting a lot of UV light, right? How deeply does that UV light penetrate into us?
Not that deeply,
but not a lot.
It doesn't penetrate very deeply, does it? It really just penetrates into our skin layer, our dermal layer, and it causes sunburns, right? That's really what's going on.
Well, but if you have an area that's exposed a lot, like I played soccer a lot, and so I got a spot of precancer on my ear that they had to take out. So
a little melanoma.
Yeah. So wouldn't that go deeper?
So yeah, what's happening there, Laura, is what's really happening when you get spots like that is what's happened is it's penetrated through the epidermal layer, right? And damaged that layer, and then it's penetrated into the dermal layer. And at the dermal layer, the cells below that start to get mutated, your keratin and your myelin cells. And so those, they call them precancerous because they're not actually cancerous yet, but those precursor cells, because they mutate out of control, they'll just remove them. And yeah, that's caused because you've consistently gotten ultraviolet light exposed to those cells.
Yeah, so they can go deep.
What's really happening is once you get cancerous cells, it's not the UV light that's causing it to go deeper. You'll get those precancerous cells, and they start to do chemical changes inside the dermal layer, and then those cells start to penetrate further down into your epidermal layer, and then they look for the blood supply, they look for muscle. Does that make sense?
Yeah, but it doesn't become cancer until it's in a little bit deeper level.
Yeah, it's got to penetrate deeper down, and those mutated cells, they start to do angiogenesis, and they start to release chemicals. They start to release cancer generating stuff because of who they are. And that's what's going on. And again, as Addison will clearly attest, you will spend a whole unit of your life in pathophysiology. And one of the things they do talk about is cancer. And they spend a lot of time talking about actually skin cancer. Exactly what you're talking about.
So fascinated with cancer.
Yeah, cancer's pretty cool. It's cool, but it's scary. Okay, so ionizing radiation, it's these high energy radiation that can go through objects. Again, we use it to sanitize and kill bacterias and for stuff that we can't put under any kind of heat. So example, like I told you guys, I worked at Merit Medical, and they do a lot of plastics like syringes and different kinds of medical equipment that needs to be sanitized. But obviously you can't put plastics of certain kinds under any kind of heat at all, right? And these are things that have been touched a lot during the process of setting these medical devices up. Like 18, 20 different people have touched the syringes that go to the hospital when they package them in the packaging and set them up. And so instead of trying to wash them and take care of them that way, there are aseptic procedures that they use to keep them clean, like covering people's hair, and everybody wears aprons and gloves and stuff. But people are breathing on this stuff. They're not have face masks on. So what they do is they keep them clean, they package them up, they put them in these big, huge crates of stuff, and they take them and run them through a particle accelerator, basically a nuclear reactor. And the nuclear reactor focuses all of its gamma radiation, X-rays, and high energy electrons through it. And just it has the ability to pass right through the cardboard boxes, right through the plastic, right through the pallet, everything. And it gets in there and kills all the funk on the syringes. And so when they get to you at the hospital, they're nice and clean and there's nothing growing on them. Okay.
Okay.
So that's it. And that's what it does. It's just ionizing, and yep, it just forms free radicals, and the free radicals go through and kill everything. So UV light, right, causes thymine dimers. And so we know what are thymine. Thymine are nucleic acids in our DNA right here. It's showing it down here. So this UV light comes through, it cuts it, and then these guys, these A's and these T's are looking to bond with somebody. They want to be bonded, right? And so what it does is it takes the sulfide bonds, I think it is, if I remember right, it's sulfide bonds. It cuts one of the sulfide bonds that's a single bond, and then when the thymine rejoins, it has to do a double bond to rejoin because one of the electrons has been blown off, went through the UV radiation, and so it causes a double bond to form to keep it stable chemically. That's what it wants to do. Okay. And so it says right here, UV light doesn't penetrate very deeply, and it's limited just to surface sterilization. So stuff deep down in your gut, in your abdomen, isn't going to get UV light hitting it and hurting it. Okay. And so what do we use it for in the medical field?
To disinfect rooms.
Yeah, disinfect rooms and water and stuff. And so like if there's water that's been contaminated with certain kinds of bacteria, we'll just subject it to UV radiation, and that should kill all the stuff in it, because light can penetrate really far into a lot of water. So any questions on that one?
No.
Okay. So again, like that would have been a perfect example. If we'd been doing like questions, there's probably 2 or 3 questions just in that slide. And if we'd been doing like kind of questions, we would have hit that slide a couple of times and known today's question. You know what I mean? It wouldn't have caught us all. I mean, even me, it caught me by surprise. And I like radiation, Okay, who's going to take filtration?
I'll do it.
Okay.
Oh, unless you want to do it, Addison.
Addison, you want to take it? Okay.
Sure. Filtration sterilizes liquids and gases without using heat. So they have the filtering liquids. They use the membrane filters, which basically catch the microbes that are too big to fit through the pores. So, yeah.
Okay.
It's basically like, would this be an example of like a water filter that you can have at your house?
Abso-freaking-lutely. That'd be a great example. What's your water filter filtering for at your house?
Clean water.
Well, most people use filter when they filter their water at their house. They're trying to filter heavy particles out of it. We're not trying to get bacterias out, right? But that would certainly be something that a water filter could technically take out, right? If the filter on it is correct, it could be filtering bacterias, right? But a lot of times here in Utah, what we're really filtering for is we're filtering for hard water. We're filtering some of the heavier calciums, some of the heavier metal particles out of our water, and it makes it a little bit more palatable, and it makes it a little bit more it makes it a little bit nicer for people's skin and hair. So it's really cosmetic what we're doing. Does that make sense? So sometimes people get itchy skin from when they wash their clothes. And the reason they're getting itchy skin is the hard water that they're using to wash their clothes. It makes their skin, it actually embeds all those heavy particles into their clothes. And so when they put them on, they're clean, but they have all those heavy particles. It makes people's skin itch and they break out into rashes. So what they'll do is they'll use a hard water filter to filter out the heavy elements. And then they'll use soft water to wash their clothes specifically, and it keeps it from getting those heavy elements embedded on their clothes and makes it so they don't itch. But for bacteria and viruses and funguses and stuff, what are we using the filtration method for?
To filter out microbes.
Yeah, we're trying to filter them out. And
big.
what's the key thing that we can do with that filter? What can we then do with that filter? We can pull that filter out and we can put it on a petri dish, can't we?
Oh, yeah.
And we can culture for those microbes and see what's actually on that filter. So we can put an unknown substance, in the filter. We can filter through it. We can pull that filter out and we can put it on a petri dish. We can leave it in 37 degrees Celsius over the week and then see what actually cultures and grows in there. And then we can like pull it apart and start to do this test to see what actually is grown on there. So that's another reason that we do filtration, is it lets us look for stuff in the lab. Okay. Filtering air. Addison, you're going to definitely take this one because we beat the crap out of this one.
Me?
Yeah, you know all about filtering air. This is one of our airborne precautions in pathophys. We spent a lot of time talking about this.
Just that we use the N95 mask. They're called that because it blocks out 95% of particles.
Yep.
We use it for action.
But yeah, we use it for what? What did you say?
Airborne precautions.
Yeah, we use it for airborne precautions and so we don't get sick. And what particular kinds of patients would we wear an N95 mask with specifically?
Tuberculosis.
Yeah, I'm on the
Um,
air with MTV, right?
yeah.
Meningitis, tuberculosis, right? And then what was the last one? Versicilla. What's versicilla?
It's not meningitis. It's
Is
right.
it measles? Is it
Yeah.
measles? I'm sorry. Which kind of measles was that again?
Rubella. I mean rubiola.
Rubiola, right? Because rubella is Mr. Pimp's girlfriend. So Laura, there's a way that there's a couple of mnemonics that they use to teach for airborne, particle, and contact precautions. And there's these little mnemonics that you'll learn, and they actually do really help to remind you what you need to wear certain precautions for. And so airborne precautions in that class it says you're on the air with MTV, and so it's the M, the T, and the V that reminds you that it's measles, rubiola, tuberculosis, and Versilla is chickenpox. Those are all airborne viruses or bacteria that you can get. And that mnemonic reminds you to wear an N95 HEPA mask and to have HEPA procedures and airborne precautions in place when you go and treat a patient with that. So that's just a little bit, it's a fun little thing that Addis and you'll hear us say that once in a while. And that's why we're saying it, because we've just been totally programmed by Tana Marchbank in the nursing program. We're her little minions and she's programmed us. Okay, so what does HEPA stand for?
High efficiency air.
Yep, particulate air. And it's used to keep biological crap out so we can be safe. Any questions?
No.
Okay, so methods of physical control. This is that slide. Okay, so it talked about heat, cold, pressure, desiccation, and radiation. So it's kind of small here. So we talked about heat. Okay, so what are the major kinds of heat that we were using in this presentation? What were we talking about?
Heat.
One requires water and one doesn't, right?
Mm-hmm.
So what's the one that was? So what were the key things about water? What do we call that when we use steam? What's the machine? We put the instruments in to steam them. What do we call that thing?
The steamer.
The autoclave.
Yeah, it's the autoclave, right? And what were the three components that I said that we had to remember for the autoclave and how they work? It's temperature, pressure, and time. So what was the temperature that we used?
121.
And then how much pressure did it need to be under?
15 pounds.
And how many minutes did we run it?
15.
Okay, so that was different than dry heat, right? Or a dry heat oven. So what was the dry heat oven conditions? What did we have to do for dry heat oven?
170 degrees.
For how many minutes?
I don't remember. 2 hours.
2 hours.
Yep, 2 hours. Yep. That's a trick question, Laura. I was trying to trick you. So it's for 2 hours. You got it. Okay. So those are the two big ones. And then we did talk a little bit about pasteurization. So Addison, what were the key things about pasteurization? What made that one special?
It's like you can do it for temperatures that are boiling, right?
Yeah, so we definitely do it below boiling, right?
Yeah. So it's like you would do this for milk.
So yeah, milk's a great example of a time that we do this, right? Because if we put milk under any more temperature than that, what happens to the milk?
It'll all curdle and get.
It goes what? I'm sorry. Say it again.
Curdle.
Yeah, it just goes bad, right? It's no longer milk, and nobody wants to drink it, right? Then you kind of ruined the point of putting it under heat, right? If you can't drink it anymore, it's what?
Cottage cheese.
It's cottage cheese. Is that what you said?
Mm-hmm.
We got to figure out why you cut out every other word.
I couldn't tell you.
We got to figure that out because it's just really frustrating because I'm like, cottage, yeah. You sound like a bad robot on Star Wars.
Whoa.
Okay, so let's talk about cold. So what were the key things about cold?
They basically they can survive but grow.
Yeah, so we
They
don't
become
actually
dormant.
kill anything, do we?
Mm-mm.
We just refrigerate or we keep them cold or get them cold, and that cold temperature takes them down past their division point where they're comfortable dividing. So they just go dormant and they just hang out, wait for conditions that are better for them. Okay. So refrigeration, what's the temperature range for refrigeration?
0 to 7.
Yeah, 0 to 7 degrees Celsius, right? And then freezing is everything below what?
0.
Yeah, the book here, the little cheat sheet shows everything below a -2 degrees Celsius, but that means everything between 0 and -1 isn't technically in a range. So I would just say that it's everything. So refrigeration would be 0 to 7, and then everything below 0 is technically freezing for water below, below water freezing point. So, but just the book says -2. Okay, let's talk about desiccation. What's desiccation? Laura, what's desiccation?
Please hold.
Think
Cheater.
dry fruit.
What?
Think vacation. Think dry fruit. D.
Dry fruit and freeze
Desiccation.
drying.
Yep. How do we get dry fruit? What do we do to it?
Take the water out.
Yeah, we just remove the water, right? And there's two ways that we can do that. We can do it by doing one way. It doesn't actually. So we do one way, it's freeze drying it, right? And the other way, how do we do it the other way? The other way that isn't freeze drying, how do we get the water out of it?
I don't know. It's just the simple desiccation.
Well, we simple desiccation is removing, just drying it out, right? Just applying it to some drier air, and then it just dries it out. But what do we call it when we put like, say, a lot of glucose around it? What do we call that? When we put a lot of glucose with, say, strawberries, what are we doing to those? When we put all that sugar around it, what are we doing to the strawberries? We're putting them into a hypotonic, isotonic, or hypertonic condition.
Hypertonic.
And by putting those in a hypertonic condition, what is that glucose molecule? What are those glucose molecules doing to that strawberry? The strawberry cells.
Sucking out the water.
Yeah, they're hypertonic, so it's pulling all that water out. Okay, we beat that one up. Okay, what about radiation? There's two kinds of radiation.
Ionizing and non.
Okay, so what's in the non-ionizing category, and how does it work?
It's UV light.
Yep, there's only one thing in the non-ionizing, and it's UV light. And how deeply does that penetrate?
Not very deep.
Doesn't penetrate very deep, right? And then what about ionizing radiation? Laura, what about ionizing radiation?
You ionize water, and it does penetrate deep.
Yeah, so it penetrates deep. So what kinds of things we want to use ionizing radiation on?
Cancer cells.
Yeah, we'd use it for cancer cells, right? Also, like medical instruments that we can't put exposed to heat, we're going to expose them to high energy electrons, high energy gamma and X-rays, and those high energy rays are going to go through and cut the DNA in those molecules, and that's going to keep them from growing. And or at least if they grow, they're not going to grow as those bacteria because we've cut their DNA all to pieces, and they get jacked up and they don't grow very well. Okay, so physical control methods. We talked about filtration. So what do we know about filtration? Addison, I think you did a pretty good job explaining this. So we had a couple of different kinds. So what was the first kind of filtration that we talked about?
It's
well, there's only two kinds of things that we can filtrate. So we can filtrate air, liquids, and solids. Out of those three things, what are the three things that we can filter?
Water.
Sorry, what was that again?
Water for the liquids?
Yeah, water's a liquid, so we can filter water, right? What else can we filter out?
Gases.
Say again. I didn't hear you. I'm sorry.
Gases.
Yeah, so gases. So we don't particularly filter out solids, right? Those we just wash those off. We don't actually filter them. We just scrub them down. Okay, so air and liquids, or gas and liquids. So How do we filter a liquid? What do we do to that to filter it?
We use that plate, whatever it's
Yeah, we use
called,
a
the
filtration
membrane
plate.
filter.
Yep, use a filtration plate to filter membrane, right? And how does the filter membrane work? How does it get everything out of there?
Well, the microbes can't, they're too big to fit through, so they stay on the plate.
So they're too large to get through there, right? And so that pulls them out. And what can we do to that filter plate, Laura, when we run that liquid through there and we get some part?
Make a culture out of it.
Yeah, we can put it in a petri dish and we can culture that, right, and see what's there. And that's exactly what we do in the lab and what scientists do to like water samples and stuff. That's one of the methods they use. Okay, what about air? What about air filtration? So, Laura, I'm going to use you this time since we used Addison for liquid. So, Laura, so what about air filtration? What was the key thing about air? Well, let me ask you a question. So you're going to go in and see a tuberculosis patient at the hospital. Are you just going to walk in there?
No, so you'd wear an N95 mask. So you do that for MTV.
So, yep, which is, you know, I'm on the air with MTV, right? So we do it for measles, rubiola, for tuberculosis, and the last one is Versilla. And Versilla is the bacterial name for what?
Chickenpox.
For chickenpox. Awesome. Okay. How else? So that's the technique that we use to protect ourselves individually. How do hospitals and labs protect people and the environment from bacteria and microbes? What system do they use to keep everybody safe?
They use the high efficiency particulate air filters.
Yep. So they use those air filters. So they basically insulate all the HVAC in the whole building with those high efficiency particulate air filters, and those things catch all the bacteria. So just in case they get out of the control samples, the control areas or the hoods, right? They also have those HEPA filters in place, and that controls everything that's coming through there and catches it all. Okay, you guys ready to stop there?
Yeah, because
we've been going for an hour.
Section if we keep going.
Yeah, there's that next section is all the chemical stuff. Okay, so listen. So let's look at this really quick. Since I'm with Laura, I think it'd be best to break this next piece up. So let's look at this really quick. So, ladies, under chemical methods of microbacterial control, there are 1, 2, 3, 4, 5, 6, 7, 8. There are 8 of them, and there are 3 of us. How do you guys want to break these up?
I'll take the first one.
2.
So if Addison, if you want to take the first, so what's 8 divided by 3? It would be what, 2 point something? Yeah. So Addison, do you want to take the first 3?
I'll take the next 3.
Okay, I'll just take the last 2 then. Okay, so that's going to get us through. Let's see what, hold on. So how many total slides is that going to get us through? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. That gets us through 17 slides. 17.
But you have to think the class period is longer than an hour.
Yeah, I know, but we have 3 days to do this, is what I'm saying, to get to the end of 13. So I'm just looking at how many total slides are in 13?
We have to do 14 as well.
There's 75 total slides in here. And right now, ladies, we're sitting at slide number 34. So there are, so that's 34 minus 75 minus 34 is 30. That'd be 31 slides. So if we each take like 11 slides, basically. So who wants to take the first 11 slides? So tomorrow, if we're going to get through all this, who wants to take the first 11? Was that in 13? We're only in 13 right now.
13, right?
What was that?
Are we tomorrow?
Yeah, what's tomorrow?
Thursday.
Tomorrow's Thursday, so it just means it's 6 like normal.
No, I just didn't know if we were going to go over 14 tomorrow.
Well, no, we haven't finished 13 yet. Is what I'm saying. There's 31 slides left in 13. If we're going to cover them and kind of incorporate what Laura was saying, that means that each of us would have to cover 11 slides tomorrow if we shared it and got through the whole thing. So that means each of us is going to have okay, so if we take an hour to cover 34 slides, that means each of us takes 11 slides, and then that gives each person about 20 minutes to cover those 11 slides. Is that fair, or does that work for you guys?
So tomorrow is Valentine's Day. I do have plans.
Oh, so... you suck.
Sorry.
Addison, Laura sucks because she's got a Valentine. The rest of us are all losers. I'm teasing Laura. I'm sorry. No, I'm happy that you have a Valentine's Day.
Thanks. Do we want to try to move it, or do you guys want to do it without me?
What do you want to do, Addison?
Well, I'm not going to move it. I don't care.
Let's do this, Laura. Yeah. Let's wish you have a good night. Let's keep it at 6 o'clock because, again, you know how it is. If we start moving around, what happens? We just start to negotiate all the time. That's not me being mean to you. I hope that you have a very, very pleasant Valentine's Day with your sweetheart.
I also do think that, like, I think we're all pretty flexible that, like, even if, like, one day, I don't know, like, I'm fine to move it up.
What I'm saying is I have to work tomorrow night, and I'm planning the rest of the day around when this study group is and when I have to go to work. Does that make sense? So yeah. I'm trying to think a little bit further past it. So yeah, no, I like I said, I'm okay. Like there are days that there are going to be days that I can't do this, you guys, because of work and other exams and school and everything else. So I'm okay with just keeping it at 6 and just going with it. And if Laura can't do it because Laura wants to go out of town this weekend or whatever too, right, Laura?
Yeah, but I'm still planning on trying to do the study groups.
Okay. So again, like things are just going to come up. And I'm just, like I said, that's why I'm trying to be not like strict with you guys. I'm just saying the more we move it around, the harder it is to plan around it and get consistent with it. So I'll let you guys vote. Ladies, what do you guys want to do tomorrow?
I don't care.
No, Addison, what would you like to do?
I'm literally good to do it whenever.
Okay.
So I think
then somebody propose a time, and then we can move forward.
You're the one that has to sleep and go to work.
Honestly, I have to be to work at midnight. So if we move it forward, it just means as soon as we're done, I'm going to go to sleep. Because I don't know about you guys, I have school tomorrow as well. Addison, do you have school tomorrow? I have school tomorrow. So if we just move it up, it means I go right from class to this, which is fine. I don't care. I finish class at 1 o'clock tomorrow. So as long as it's after 1 o'clock, I'm okay.
Yeah. I just think that, like, I think as long as we all just, like, schedule it and be like, let's meet at this time, then I'm good with it.
Okay. Like I said, my thing is I like to just get it on the calendar and be consistent. It's just easier to be consistent to get ready, too. Because, like, now I got to be ready with 11 slides for tomorrow, right? Or whatever we're doing. And again, I've got another. Does that make sense what I'm saying? Like, we're all planning around everything else.
I don't want to stress you out. So why don't you guys just keep it at 6 and I'll go over this by myself. Okay?
Okay.
All right. Because you're stressing out about it. So you guys.
No, I'm stressing out because I got 2 exams I still have to study for.
Yeah.
We're moving this thing forward. We're moving this forward now. I have class tomorrow from 8:30 in the morning till 1. And that gives me, if I'm going to get ready to be with you guys tomorrow for this, I've got to do it right now. And that's cutting into studying time for other stuff. Is that fair, or am I just being a jerk?
You have 2 exams tomorrow. So
yes.
If you want to just not do it tomorrow, that might help you not freak out so much.
Well, no, I want to know that if I'm not doing it, you guys are. That's the whole point of trying to do this, that I can't be the ringleader all the time. I love being the ringleader. It's awesome. I love doing it. But I like to think that when I'm not doing it, you guys are doing it. Does that make sense?
So would it be easier for you not to do it tomorrow?
Well, that's fine. I like I said, I'm okay with it. I'd like to know that when I'm not physically part of it, just like when Laura, you can't be here, you know that Addison and I are doing it. And vice versa. When Addison's got to work, she's confident that we're doing it without. That makes sense. The point of it is that I like the study group to be bigger than one person. And if that's not the case, then I just am flattered that I'm the ringleader, and it's awesome. I'm totally flattered I'm the ringleader.
So
okay, listen.
The more that we can all work together for more days, I just feel like that's better. So I'm good to do it whenever, but it's
okay. Listen, this is what we're gonna do. I am going to excuse myself from tomorrow's. Addison, you and Laura clearly can communicate with each other. If you guys would like to meet and can coordinate it, I would think that would be awesome. And I am going to get off the horn and go study for my other exams and not worry about it tomorrow night. And then on Friday at 1 o'clock, I'll be ready to go again.
Okay.
And if you guys decide to like give up slides or divvy things out to study, just let me know tomorrow after you guys get done. Say, hey, you're responsible for unit 14, slides 1 through 10 or whatever. And I'll say, yeah, I got it. And I'll be ready that day. I'll be ready Friday with those. Is that fair?
Yeah.
Mm-hmm.
Is that good with you, Addison?
Yeah.
No, this is just the growing pains of every study group. It's always like this. There's always some growing pains, and you're dealing with people, so you got to kind of get along. And so I'll step away, study for my other exams, and let you ladies have a good evening, and let you guys figure out what you're going to do tomorrow. And then I will just, yeah, I'll be fine.
Okay.
Okay. Thanks, ladies.
See you Friday.
Have a good night, guys.
Okay.
Okay, bye.
Too.
Bye.